Today, we're going to explore a provocative framing of insulin resistance, not as a flaw, but as a built-in cellular defense system against oxidative stress.
Why pick these papers is that in our diet protocol, one of the things we say is no seed oils. What's interesting, what I find when I go online and I see all the seed oil talk, anti-seed oil talk, is that everyone will talk about inflammation. People will talk about how they process the seed oils. They'll talk about the omega-6 to omega-3 fatty acid ratio as being skewed, but no one's talking about reactive oxygen species.
I think a lot of people think of it as oxidative stress. ROS are bad, and they absolutely can be and absolutely are, depending on the level of production in the cell and the mitochondria, that a lot of ROS effectively can, for lack of a better term, blow up the cell. Necrosis. It also signals apoptosis. All things can go haywire. But also, there are levels of ROS that they sort of fluctuate between, you could call low physiological and high physiological, and the in-betweens. Work as signaling molecules, and they're not just sensor or a gauge. They're effectively like a switch, so they can actually modify the structure and function of proteins. So it's not just like this, phone call, hey, something's going on. It actually acts upon particular proteins.
I think that's how you explained to me originally, like why it's so important. It is of the first order in terms of the cellular mechanisms in response to like energy demands of the body.
Yes, this little picture it's showing, it says reactive oxygen free radical O2 minus, and that's another word for that. We'll get into it, is superoxide. There's a bunch of SOD abbreviation in this, is superoxide dismutase, which is like an endogenous antioxidant that converts superoxide to something called hydrogen peroxide. You might have this in your medicine cabinet. Oxygen is called a terminal electron acceptor. And so what we'd say normally, it's difficult because it's not abnormal to create ROS, but they're always created at some level. This is also the electron transport chain. And this is just to show, if you see in red, those O2 dot minus, those are spots where sites of reactive oxygen species, and in particular, this is superoxide. So this is mitochondrial superoxide. So MSOXP is mitochondrial superoxide production on the top. Effectively, people are saying, what causes insulin resistance? It depends on who you ask. And I think Schulman would argue, it depends on what you look at. It depends on what you're studying. Are you like taking a step back and looking at everything? Or are you in the weeds looking at inflammation? And the reality is like, the answer you could say is yes to all of these. Like all of these things in culture, in an animal model, you can induce insulin resistance. And interestingly, I didn't pick the slide or figure for it, but you can see upstream of that too is ectopic tissue fat. Mm-hmm. You're looking at like a lipid overload. So when people say like cancer is a multifactorial and it's complex, it's because it's a marker, it's a proxy for how little we understand, is how I would argue. And so I think that this is my bias, but I think these guys are very clever in what they do. They're trying to look at like, okay, what's upstream of all this? And when we talk about insulin resistance, because the way we study it, it's typically the muscle's response to glucose uptake. But there's something called NIMGU, which is non-insulin mediated glucose uptake. And that is through muscle movement, mechanical movement, fight or flight, insulin resistance. No, don't I want fuel? That's no problem if you're running, right? You don't need insulin in order for the muscle cells to take up glucose and fatty acids. What do corticosteroids do? They flood the bloodstream, and so through diffusion to the cells, is that they flood the bloodstream with fuel. And so you have all this fuel available. Are those four models associated with, when I say MSOX, which is a form of reactive oxygen species production? We'll probably gloss over the difference between superoxide and hydrogen peroxide, because it's not particularly, well, these guys probably argue it is important, at least at this time. It's called translocation. They move to the surface of the cell. And those are transporters. So it's saying, let the glucose in. And that's how insulin works with letting glucose in. Something that's not mentioned a lot, but I'm going to have to mention it here, is that they also can bring in fatty acids through something called CD36. Yeah, the more fat that they give the cell, the more insulin resistance they see, which they're using a glucose transporter as the measurement, which I would say it's a tell, but that's what we do. We look at glucose. We don't necessarily look at fatty acids. Superoxide production is a common feature of many different models of insulin resistance in adipocytes, myotubes, and mice. It's a spoiler alert, because they're also going to look at mice. And so the next thing that they did after they saw that there's an association here, what if we inhibit just the superoxide? Will that reverse the insulin resistance? If you look at the amount of reactive oxygen species that are created in a cell, it's predominantly, I don't know if it's like 90 something percent that ROS is created in the mitochondria. Now they're looking at, okay, we looked at the fat-induced one. just like they've looked at the inhibitors, the mitochondrial inhibitors. But in this case, they're looking at muscle cells and fat cells, myotubes and adipocytes.
I don't know if this is normal.
Well, my bias is it's somewhat typical depending on the journal article you read. I think it's really important. There are a lot of like typical meaningless, but like if you read a Nature paper and it's an experimental study, they're really cool, even if they're meaningless, whatever it is, because it really is like an investigation, and it's like a journey. If it's done well, where they see something and then they go, is what we see what we really think we see? So let's try to rule out this. Okay, do the findings hold up in vivo? So all of those previous findings, they're looking at myotubes and 3T3, L3T3. They're looking at, often never really put it this way, that I think with the Ross hypothesis, we often put it that way. It effectively looks like the cell is more hungry. That it's saying, feed me. It's more sensitive to taking up glucose in the cell. Rightfully so. A high-fat diet is almost by default. If you're not messing around with a protein, it's a low-carb diet. So is this a low-carb diet? This is a base that says ketogenic diet in mice. It was Megan Roberts, if you want to look at like a study from somewhere like around 2018, that increases median lifespan in mice or something to that effect. You could call it a high-fat diet, but this a 90% fat, virtually devoid of carbohydrate diet, 10% protein instead of 20% protein. You could argue maybe there's protein that's involved.
So this doesn't induce obesity, this one. This is not an obesity-inducing diet. We had the other two, the typical one and then the one that these guys used.
Yep.
And the ketogenic diet isn't advertised in research diets as a obesity-inducing diet?
I think it's just advertised as like a ketogenic diet, not a diet-induced obesity model. I don't think it would be in that category.
To be thinking about not only the previous in vitro experimentation, but also the hypothesis that ROS is a cellular defense mechanism against nutrient toxicity, essentially, right? This insulin resistance.
It's a physiological signal, and the response to like a high-fat diet is the mice are, whatever you want to call it, there becomes like pathological complications. If
you have the bias in thinking that just all insulin sensitivity is good and all insulin resistance is bad, just the in vivo experiment in the mice, then right away your interpretation of the results is going to be that fat is bad and antioxidants are good.
Yes.
Right. So that's the bias that people bring to these things without looking, potentially upstream. Like, well, and we'll get more into the Ross hypothesis, but yeah.
This is Ron Mittler. This is that ROS are good. And I think he gives a good overview with his figures. We went through this a little bit, that these ROS are primordial. And this is worth looking into as well if you want to look at when we talked about how can the reactive oxidant species actually alter the structure and function through its signaling. It's absolutely beautiful because it effectively opens up the insulin signaling cascade. It says, feed me. And so what happens? You feed the cell glucose and fatty acids. What happens to those? Predominantly, you're feeding your mitochondria. Your mitochondria starts using it. So it's filling up on ATP. And as it fills up on ATP, it's building up the membrane potential. If we go back to that, it's reducing CoQ, particularly if it's a high fat. That's a loaded term, high fat diet. But say that you're eating a lot of saturated fat along with it. Actually, if you give it a little burst of ROS, it's a really nice way to increase the insulin sensitivity.
When people are looking at this, it's important to think back to what we were talking about before with the nuance of insulin sensitivity and resistance at the earlier stages. Like you said something profound, which is like, if insulin is going to trigger nutrient ingress into my fat cells and I don't want to get fat, what I want is a little bit of insulin resistance.
If somebody fasts for a week, you're inducing insulin resistance. And part of that is you're sparing glucose for the brain, you could say. But the brain is predominantly using glucose.